When a person inhales cigarette smoke, the nicotine is extracted from the tobacco and is transferred in the form of smoke particles to the lungs, where it is quickly absorbed in the pulmonary circulation. Then, it enters arterial circulation and it is quickly transferred to the brain. Nicotine is easily dispersed throughout brain tissue, where it binds itself to the nicotine acetylcholine receptors, which are ion channels. When nicotine binds to an ion channel, a change in its structure is happening that forces the channel to open. As such, positive ions enter, causing membrane depolarization that results in release of neurotransmitters. The neurotransmitter is transformed to an electrical signal through an ion channel in an adjacent neuron. The secretion of neurotransmitters mediates the manifestation of various effects of nicotine, such as pleasure, relief from anxiety and tension symptoms, appetite suppression and memory enhancement. However, prolonged use and abuse of nicotine leads to addiction. Cigarette smoke, though, contains many harmful substances. Prolonged smoking in order to receive the addictive nicotine causes extensive damage to almost every organ of the human body. Nicotine decreases skin temperature and increases blood pressure and heart rate. Smoking causes dyspnea, fatigue and increased anxiety until the next cigarette. In the long run, smoking is a major cause of cardiovascular disease, cancer, especially of the respiratory tract, respiratory problems such as chronic obstructive pulmonary disease, as well as other non-fatal diseases. However, most of the damage caused by smoking is reversible when a smoker stops smoking. Different approaches exist to help people quit smoking. One of them is non-pharmacological treatment. The Smoke-Free Brain framework has developed a multiple-visit program that incorporates biofeedback and neurofeedback for smoking cessation in young unemployed, asthma and COPD smokers. In the first visit, a pulmonologist takes an extensive medical history in order to decide for the subject's eligibility to enter the study with regards to inclusion/exclusion criteria. Then, a pulmonary examination is carried out through spirometry and asthma provocation in some cases. Exhaled carbon monoxide level is measured along with blood sampling. Finally, the participant fills questionnaires and behavioral tests regarding the degree of nicotine dependence, the health status, as well as different quality of life parameters. During the second visit, each participant undergoes a baseline recording of brain activity by means of high-resolution electroencephalography. The participants during the next 5 visits do biofeedback training to raise limb skin temperature. The procedure for each biofeedback entails the participant comfortably lying on a recliner looking at a screen 2 meters away, connected to a temperature sensor, while behind him the trainer oversees the session and guides the participant. When the participant is able to relax and raise the temperature of his limbs, he watches a puzzle solving itself and listens to relaxing music. Whenever he is not relaxed, the music stops and the puzzle freezes. When the participants complete the 5 biofeedback sessions, after 3-4 weeks, they return for an intermediate evaluation, including a clinical examination and another EEG recording. After the intermediate evaluation, the participants undergo neurofeedback, to a total of 20 sessions. In each session the participants are lying on a comfortable recliner in a similar setting as before and are connected to EEG electrodes. A cap with a low number of electrodes is worn, while the earlobes are prepared with alcohol and connected to a ground and a reference electrode cable, while an active one is connected to the PZ site. The participants are instructed to relax as much as possible, almost reaching drowsiness. On the screen, 2 bars correspond to alpha-theta ratio and to theta-alpha ratio. The goal is to increase their theta-alpha ratio. When the participants complete the 20 neurofeedback sessions, they return for another evaluation, including a clinical examination and another EEG recording. For the following 3 months, the participants do not take part in any intervention, and after that period they return for a final examination and EEG recording. After 5 sessions of biofeedback, total oxidative stress of the participants was marginally reduced compared to before, while 6 out of 10 participants presented a significant reduction. Also in 5 out of 27 participants in total, the levels of exhaled carbon monoxide were reduced below the threshold of 8 parts per million, that is considered able to distinguish smokers from non-smokers. 4 participants reduced their addiction to nicotine from moderate level to low, and another 4 participants reduced their addiction level from high to moderate. In total, participants scored significantly better in general health test, considered a screening tool for psychological disorders. Moreover, the participants reduced, non-significantly, their depression and anxiety symptoms following the 5 biofeedback sessions. The default mode brain networks of the participants did not present significant alterations after the biofeedback training, either in their connections or in their properties, like density, characteristic path length, etc. This was interpreted as a possible neuroprotective mechanism of the biofeedback. Nonetheless, outflow of information from 2 brain regions of interest, right ventrolateral prefrontal cortex and right temporal pole cortex, was marginally significantly greater after the biofeedback training. These 2 areas are considered to play a role in inhibition, in control of addictive behavior, and in certain aspects of memory and emotional processing. Following 20 sessions of neurofeedback, significant improvement was shown again in the degree of nicotine addiction, in self-esteem and in sleep quality as well. Women in particular showed greater improvement in self-esteem and in anxiety traits. As with biofeedback, no significant reorganization of default mode brain networks was observed after neurofeedback training as well. Again, this is interpreted as a neuroprotective action. Participants did present, on the other hand, greater inflow of information in the region of left medial prefrontal cortex,